Thank you everyone for joining us today. My name is Bill. I am the lead for the GDC User Services team, and I'm here with my colleagues Zhenyu and Chin. I'll let them introduce themselves at their sections. But today I'd like to talk to you about GDC copy number variation analysis, workflows, and tools. Um, just to go over a few webinar logistics, like Sharon said before, this webinar will be recorded. We will make the recording available on gdc.cancer.gov. We will need to process a little bit to add captions and make it 508 compliant, but after that, we will make it available. The recording and the slides both will be made available. And if you have any questions during the webinar, please feel free to type it into the Q&A panel. We will get to questions at the end, and we'll also allow you to ask questions at the end too. So feel free to save it for the end or type it anytime you'd like. So just to go over the different sections we're going to talk about, we're first going to go over the harmonization workflows and the files. That'll be Zhenyu and I. That'll be followed by a demo of the GDC copy number segment tool. After that, we will be doing a demo of the GDC CNV API. And after that, we'll address any questions you have. So that being said, I will pass it over to Zhenyu, who will start in this section.
Thanks, Bill. Good afternoon, everyone. This is Zhenyu, and I lead the bioinformatics team in the GDC. Today, I'm going to give an overview of all the copy number variation workflows we have ever used in the GDC. Next slide. Over the years, we have built and supported multiple pipelines. There are two main reasons for so many pipelines. And first, we have used two very different strategies. One being the arithmetic SNP6 array used in early projects like TCGA and Target. And then later we moved to project with whole genome sequencing. And a second reason of that is the somatic copy number inference is really a very difficult problem, even up to now. Tumor samples vary very widely in purity and ploidity and clonalities. So a highly pure tumor with subtle copy number changes can look very similar to a less pure sample with drastic CNV changes. This is why we created several workflows to get the best possible results for everyone who uses GDC. And I will go through each of these workflows in roughly the order we introduced them in the GDC. Next slide. Next view. The first workflow was called DNACopy. This one was used only for TCGA and Target, and is based on the SNP6 data as mentioned. The input is not the original raw cell file from the SNP6 array, but instead it's the intermediate call product called tangent copy number files generated by the TCGA working group using Bursuit. The workflow produces two main outputs, one called copy number segment, and the other is called masked copy number segment, both of which are log-scaled segment means values, so they are not integer values. We strongly recommend using the masked version because it filters out probe that frequently affected by genomic copy number variations. So this is a file with more reliable results. The unmasked version is still available, but provided for historic reasons because the original TCGA dataset contains both data files, but the masked one is the one we recommend everyone to use. Next slide. Then we move to a new algorithm called ASCAT2. Unlike DNACopy, ASCAT2 uses the raw SNP6 cell file as input. It also generates slightly different output. The segment it generates is the integer value we call it absolute copy number segment instead of the log2 segment means. Besides this allele-specific copy number segment, we also generate a gene-level copy number estimate. Bill will give more details on what exactly is in the allele-specific copy number segment, but you will see this output being consistently generated from most of the rest of workflows I will introduce later. These files let you know how many exact copy numbers each gene or each segment has. And there are non-negative values. 0 means complete homozygous deletion, 2 means diploid, and anything above 2 means copy number gains. Next slide. The next version of ASCAT is released. We also adopted it. For TCGA data, the author of this tool, the Peter Van Loos lab, actually have already generated the output in HD38. So rather than rerun everything, we reformatted the output they provided and integrated these results. The output format is exactly the same as ASCAT2, just with a slightly updated algorithm. Next slide. We also integrated some results from a new workflow called Absolute. This is a tool developed at the Broad Institute many years ago, and is still one of the classic tools everyone is using. For this, we also didn't rerun the tools on the raw data. Instead, we used the segments already published from the TCGA pan-cancer studies. The reason is this data has undergone significant manual curation by community experts that we couldn't easily reproduce in the GDC. So we rely on their curated HG19 segments, lift them up to HG38, and produce the gene-level copy number estimate. After this SNP6-based approaches, we moved into the whole genome sequencing. Next slide, please. We initially used ASCAT NGS. You can see it's called ASCAT because the algorithm is very similar to ASCAT 2 and 3. This tool takes paired tumor and normal whole genome BAMs and produce very similar output as the other ASCAT workflows, including the allele-specific copy number segments and the gene-level copy numbers. The pipeline is currently deprecated, but the files are still available in the portal. The next slide. The ASCAT NGS workflows is replaced by the combination of GATK4 CNV and whole genome absolute workflows. The GATK CNV workflow also takes paired tumor normal BAMs, and the output include segment mean values and an auxiliary file that contain many detailed intermediate analysis results, including germline allele frequencies and a lot others. This file is designed for export for manual review purpose, and we don't expect regular users to consume this particular file. Next slide. The segment mean segment generated from the previous workflow, GATK4 CNV, was then fed into the whole genome absolute workflow to model purity and ploidy. This workflow generates a PDF auxiliary file with multiple potential purity and ploidy models. Among all these options, the first model was then chosen from all these potential combinations and then used to produce one set of integer copy number allele-specific segments and gene-level copy numbers. That's why the workflow includes the word auto in its name. So the combination of GATK4 CNV and the absolute together generates the integer value copy number segments, the segment mean value copy number segments, and the integer value gene level copy numbers. Next slide. To wrap up, let's look at everything together. I know we have talked about 7 workflows in total, and 4 are based on the legacy SNP6 data, and they are only available to TCGA and sometimes TCGA and target together. 3 are based on whole genome sequencing, so they are widely adopted across the GDC, including TCGA and target, and many of the other newer projects. For each strategy, one workflow produce copy number mean segment mean values, and the rest generates integer values, copy number segment, and gene level segment estimates. Right now, 2 of the workflow labeled here in red, GATK4 CNV and Absolute, are the only 2 currently active and maintained in mass production. Okay, that concludes my talk. Thank you for your attention. Please type your questions in the chat, and I will pass this over to Bill.
Thanks, Zenyu. And now I'm going to go over a little bit about the files that Zenyu was just talking about and how they are formatted. So a lot of the files do have the same format, so I kind of squeezed them all into the same section here. So ASCAT 2.3 NGS absolute, the allele-specific copy number segment files have these are the columns that they have, so I just kind of listed them like a list. So you're going to see a lot of common elements between them. In this one, we have the aliquot UUID in the GDC. Of course, the chromosome number and the start end of the segments. Those are always important. And skipping over copy number, I'm going to go first to major copy number, which is the copy number that was determined using only the larger strand, and the minor copy number, which is the copy number determined using the smaller one. So the copy number value is the sum of both of these together. Going to DNACopy and GATK4, the CNV segment with the segment mean file, there's a GDC aliquot column just like the other one, except for DNACopy, it's the aliquot UUID, but for GATK4, it's the aliquot submitter ID. So it's like the TCGA barcode or the barcode for whichever project you're looking for. This is followed by the chromosome and the start and end, common things there. The number of probes is the next column, and it's the number of probes that support the segment for DNACopy because this is a SNP6 array-based. It's a SNP6 pipeline. So you may be saying, okay, GHK4, that's a next-gen sequencing pipeline. But what I will say is that the number of probes being higher is always equal to more support for the segment. So a higher value for this column means more support for that particular segment across all of the pipelines. Then we have the segment mean, which is the log2 copy number divided by 2. And this is this way so that diploid regions will have a segment mean of 0. Amplified regions will have a positive value, and regions with losses will be negative. So 0 is kind of the baseline for diploid, and positive is amplified, negative is losses. Then for the copy number estimate files, which are the gene level copy number, these are the ones that show copy number by gene. So first we have the gene ID, so that you know what gene it is, followed by the gene name. That's the more human readable symbol. The gene ID is the ensemble ID, so it starts with ENSG and then a long number. Then it's the chromosome number, start and end, just for where the gene is in the genome, followed by the copy number, and the copy number is the weighted mean between the min copy number, which is the minimum value over the overlap segments, and max copy number, which is the maximum value of the overlap segments. So the segments all have a copy number value and they overlap the genes, so it gives those values. And then the regular copy number is just the weighted mean of these two, of all of the overlapped regions. So then lastly, I'd like to talk about the copy number auxiliary file. This is produced from the GTK4 CNV pipeline. And this is a group of different files, intermediate files, and this includes things like germline allele frequencies. And it's, like Zenu said, to be consumed by experts. So most users won't be downloading these, but if you are interested, they are available, but they are controlled access. So you will need to download, or you will need to have dbGaP permission for whatever project you're looking at to download that. So then this is also produced from Absolute GTK4 CNV Auto, and this is a PDF that contains all the potential CNV models so that they can also be analyzed. So some other copy number derived data that I'd like to bring to your attention actually falls outside of files. So I put them in a separate section here. But these are values that are on the copy number segment file. And this includes the tumor purity, which is the ratio of tumor cells to total cells, and the tumor ploidy, which is, of course, the ploidy, or the number of chromosome sets in a tumor sample. These aren't actually, if you download the file and you open up the file, you won't see these values. These actually exist as metadata on the file. So files can be filtered by these values, or you can download them as metadata, like you might download case and aliquot values or sample information. So that is all available on the GDC portal and API. So that's the end of this section. Now I'll be passing this over to Shin, who will give a demo on the copy number tool. So over to you, Shin.
Thank you so much. Hey everyone, this is Xinzhou from Senju, and it's my great pleasure to demo the copy number tool that my team built for GDC Portal. So to launch the GDC Portal, you should go to the Analysis Center, and then find this card labeled copy number segment. You can either use the demo button to look at the demo or use the play. So I will click on the play button. So by default, this tool will show a search box to ask me to search for any gene or region in the human genome. I will type PAX5 to search for the PAX5 gene in the human genome. So this is a copy number tool that we're showing. This tool runs in a genome browser in there, and so first of all, I will talk about the general navigation of the genome browser. To zoom out on the genome browser, you can use the navigation buttons on the top of the genome browser. I will click on X2 to zoom out. Now, to zoom in into the genome browser, you can either click on the in button or drag on the genomic ruler over here. So I will press cursor on the genomic ruler and drag over this range to zoom in. So now I zoom in and reduce the flanking size of the PAX5 gene. So the CNV tool, in this genome browser, there are two genome browser tracks. The first one is a CNV tool showing all the CNV segments from the current cohort. Second one is gene code track showing all the transcripts. In the CNV track, each line is one CNV segment with a color corresponding to the CNV change. We're showing the categorical CNV here. You can scroll down to the bottom to find the legend telling you there are 4 CNV changes and the color for each. All right. So you can hover a segment to see the details of this CNV change. And also you can click on one segment to get additional detail about the case, about the sample that harbor this CNV change. You can click on anywhere else in the portal to dismiss this panel. So the CNV segments are grouped by cases, and then each row is one case, and will contain one or multiple CNV segments from this case in a view range. The cases are sorted by the start position of the first CNV segment in ascending order. This pattern can be seen better by reducing the number of CNV segments shown. To do that, there are multiple ways to reduce the number of segments. I will use the segment length filter option, which is available on the bottom right of the on the copy number on the segment on the category legend part. So we'll click on this prompt, and it shows that by default the CNV tool is limiting the segments by under 2 megabases. I'll change this cutoff value to 80, and then press enter. So now you see that as indicated on the left, these labels, the number of CNV segments has been reduced to only 90 segments. And then there are 76 cases. It also shows that some cases has a multiple. It's better seen that this case has a multiple CNV segment from this region. So now I will relax the cutoff from 80 kilobases to 200 kilobases. So it now reveals a recurrent focal copy number loss events within the PAX5 gene body, as this is indicated by this stack of green lines. Each is a deletion event from a tumor sample. This is a known PAX5 introgenic deletion hotspot. There are also amplification events. These shorter red lines, the red line do not coexist with the green line in the same rows, meaning these amplification events are from a separate set of samples. To better see them, I will use a legend to filter to only show the copy number gain or amplification events. So scroll down. I will click on the loss legend and click on hide, and click on the homozygous deletion and hide. Now back to the copy number to track. Not only the pink and red lines are shown representing the gain and the amplification events. We'll clearly see that these recurrent amplification events, they are all centered on a few PAX5 exons. These represent the PAX5 internal tandem duplication events that drive a subtype in the B-cell leukemia. To observe the additional CNV pattern over PAX5, I will turn on the visibility of a loss and deletion. So I'll just click on the strike off item from the legend. So now they're back. I'll also change the. I will zoom out to show the neighboring gene of ZH7. I will drag on anywhere on the genome browser to PAM. So I will also change the maximum segment length cutoff back to 2 megabases, the default value. So it now displays a second deletion hotspot, as indicated by this second stack of green lines. They appear, also worth noting, they appear as 2 separate stacks, meaning that these 2 deletion events rarely co-occur. So the CNV tool can help us find out what cancer types these deletion events come from. The following feature, they're designed the same way as a lollipop plot, already in the GDC portal for exploring the coding mutations. So on the left of the CNV tool, there you can, under the track label, you can find this 500 samples label over here. I will click on this label, and it shows, now you see show a panel of summary of disease types, with the acute lymphoblastic leukemia as the most recurrent one. I can also switch to the primary diagnosis, and I can see that the most recurrent primary diagnosis is the common precursor BAL. So all these items are clickable. I will click on the common precursor BAL, and now it will now it creates a subtract for those GDC cases from the common precursor BAL group, and in which you can see that the 2 deletion hotspots that we observed before, and they're mostly from the common precursor BAL. Again, from this subtract, I will click on this its own samples label, and now it shows and then toggle to the age of diagnosis. We can see that from this violin plot distribution, most of the cases are childhood. We can click on the close label to dismiss this subtract. So finally, we'll zoom out to explore the CNV profile at a bigger genomic range, even at the chromosome level. Before we do that, I will highlight the Pas5 gene body region, so that when we zoom out to the very big region, we know where the Pas5 was. Now I will zoom out tenfold into a 5 megabyte region, and the CNV track now change the rendering style into a density view, with densities of gain and loss shown separately. The y-axis is number of over here. The y-axis is number of events. Number of CNV segments is now over 1,000, as shown on the left. When this number exceeds 1,000, the CNV tool will switch to the density view. The highlight, this highlight box part points out where the Pas5 is, and it clearly shows the enrichment of the deletion events. So we'll zoom out. We'll continue to zoom out. Click on the tenfold again to zoom out to a 50 megabytes region. It now displays, in addition to the Pas5 region we observed before, it now shows a region with much stronger deletion event compared to Pas5. This is the significant 2A2B locus, and so if we want to, we can always zoom in back to this second deletion hotspot, much stronger deletion hotspot, to observe its details, and we can see the gene annotation on here, and we can also click on the samples summary, and we find out that they are mostly the brain tumor gliomas. So if we click on the gliomas, it will show it will limit the number of CNV samples down below the 100 cutoff, and now they're all shown at the lines, and we can see the details of individual deletion events. So that's the end of my demo. Thank you so much.
All right, thanks, Shin. And let me go ahead and get back to the presentation here. And I'm not going to go into the presentation mode because there's just one slide I need to show before we get into the demo. And that's this one here. So for the copy number variation API, there are 2 sets of copy number endpoints. So there are CNV and CNV occurrences, which provide information about specific gene level CNV mutations. So this is a gene has an amplification kind of information there. And then for the segment CNVs and segment CNV occurrences, those provide information about specific CNV segments. So those are based on segments, not genes. So another thing I want to bring to your attention is how the CNVs are classified. Our previous classification had only two different values. There's gain and loss, and I put neutral in here because of the 5 category one. So the 5 category is our newer classification. It has amplification, gain, then neutral isn't there, but that's what we count in our count of 5, and followed by heterozygous deletion and then homozygous deletion. It goes from higher copy number to lower copy number here. So I'm going to go ahead and bring up some other slides here. On the left, we're going to have some slides. And then on the right, we're going to do things on the terminal to demo the API. So the API can be used with other tools such as your browser or Postman, or I know there's lots and lots of tools to use API, but today we're going to use curl on the command line. But a lot of the things I talk about can be ported over to other softwares too. So starting out, I want to go over the 4 endpoints here, and kind of give you the difference between the CNV endpoint and the CNV occurrences endpoint. So CNV endpoint points to specific mutations and not as they apply to a person, just as they exist. So one CNV would be an amplification in TP53. That would be a CNV. But CNV occurrences are these CNVs, but as applied to cases. So if case A has an amplification in TP53, that is a CNV occurrence. So the difference between these is these are related to specific cases that have them. And it's the same thing with segment CNV and segment CNV occurrences. Segment CNVs are just the mutations, and then occurrences are how they relate to cases. So we're going to go through this demo with a series of questions or tasks we're looking to do, and they'll be in the circle here. So the first question is, how many amplification CNVs are present in chromosome 17? So let's look at our file names real quick. So first I want to bring to your attention, I put together this this payload file. It's in JSON format, and we have one query. It's we wanted to look at the amplification CNVs. So CNV change 5 category equals amplification, and chromosome will equal 17. So here, this is how the filters are formatted in the GDC API. You can URL encode them if you want to, and just put them directly into a browser. But here we're going to pass the JSON file directly. So what do we need to run a query? Just to go over the basics for the GDC API. And we do have previous webinars about the API if you want information specifically about that. But just to go over real quick what one needs to do so. First, I'm going to run using curl. So that comes on, I think, most Macs or probably Linux machines, but you can download it pretty easily. And then we're going to do the request type post because we are providing a file instead of including it in the URL. Following that, we're going to use the -h for header flag, and we're going to put content type :application JSON. And that just tells curl we are going to use expect a JSON file. Then we're going to use our URL, which is api.gdc.cancer.gov. And that's what all of the API endpoints start with, of course, /CNVs. So now we have that ready, but now we do want to give it the payload file. So we just pass the data flag, and then we use the @ symbol so that it knows to look into the file instead of looking at this file name itself. So we can use CNV payload.json, which is the name of the JSON file I just showed you. And then we're going to go ahead and run this. And as you can see, it's a little messy. So let's make it easier to read here. And one way you could do this, if you're just interested in JSON formatting, is if you pipe it to something like JQ, this will work. So if you use JQ, you can use that. Or alternatively, we do, in most of our endpoints, have pretty equal. We wouldn't pass it there. You would put into the payload pretty equals true, and that would format this in a nice way. So now we're going to pass a different payload here that so CNV payload 2, and that we're going to pass it, give it a few things that make it easier to read. So under the filters key here, we have this JSON. It's the same one as before. Chromosome 17, 5 category change, amplification. But we want to give it a bit more information. So the next thing we're going to give it is size. So if you just use the GDC API with almost every endpoint, it'll only give you 10 of the 10 of the results. So you'll need to change size to get all of them, if it's higher than 10, of course. So what you can do is see how large the full set is, or just use a number you're sure will be larger than the full set. Then format equals TNV or TSV to make it, parsable in an Excel spreadsheet or something like that. And then let's say for the fields, we didn't want all of those that were provided in the default, because before I didn't provide the fields um, uh, parameter. So let's just say we want the chromosome, the start position, and the end position. So I am going to get out of this, and I'll just press up a few times to get us to the previous API call we did. So all of the things that I give it are going to be the same, except we're going to use CNV payload 2. And I'm going to remove this JQ parameter because this is going to be a TSV output and it won't work. So I'm going to go ahead and run that. And as you can see, we have just a bunch of columns here. Actually, I made that really large. So let's go ahead and do that again, but pass it to less so that we have, so we can see the column names. So as you can see, we have chromosome 17, start position, end position, and ID. So here are a bunch of the CNVs here. So getting to the next endpoint is CNV occurrences. So of course, this is the same as the CNV, but as it applies to cases. So the next question I want to ask is, in how many and in which cases do amplification CNVs occur in the gene TP53? So the query here is going to be similar to the last one, except it's going to be amplification, and instead of pointing to a specific chromosome, we're going to point to a specific gene. So gene.symbol is TP53. Let's look at the CNV-O for occurrences payload. And very similar format here. Here's the filters. CNV.consequence.gene.symbol is TP53, and CNV.CNV change 5 category is amplification. Then we're also going to add a size parameter right away. We're going to format it in TSV, and for the fields, we're going to add case submitter ID for case to identify which cases have this mutation. The primary site and disease type, which gives us where the primary tumor is anatomically and which type of disease it is, and followed by the project ID. So if you're interested in if it came from TCGA, Target, CPTech, you could see it there. But the last thing I want to point out in this example is these field names. They might seem like you wouldn't know what to say exactly for symbol. Like if you just put symbol here, it wouldn't work. You need all of these prefixes. And how you determine what these fields are actually called is to curl the endpoint. We'll do CNV occurrences slash and do underscore mapping. And unfortunately, this doesn't have a pretty parameter, so what you have to do is feed it into a JSON formatter or copy and paste it. And what you'll see is all the information about the endpoint And if you look under the fields category, here are all of the possible fields here. So these are you would copy and paste this into your query. So if you wanted to see what symbol was, here it is: CNV, consequence, gene, symbol, and that's what we put in our payload. So let's go ahead and run the payload, but there's a few. We're going to use the same query, but we're going to make a few different changes here. One is we're going to change the payload, of course, to the CNV occurrence payload. And then we don't want to use the CNV endpoint. We want to use the CNV occurrences endpoint. And go ahead and run that. And let's we'll make this a little prettier just so that each line is on an actual line visibly. But we have disease type. So you could see things like adenomas and adenocarcinomas, squamous cell neoplasms, etc. The primary sites, so you'll see anatomic sites there, pancreas, bones, bronchus and lung, all of that. Projects look like this. And of course, this would look much better in an actual spreadsheet because all the different fields are different lengths, so the tabs are put in weird places. But here we have case.submitter ID, and these are the actual case IDs. So you've probably seen these before. This just represents one case, and these can be all tallied up. But as you can see, we're down to we gave it a size of 5,000, and we have 132 lines, so that's 131 examples of cases having an amplification in TP53. So going to the next endpoint, we're going to use segment CNV's endpoint. And for this example, we want to provide the segments in chromosome 2 that are at least 100,000 base pairs in length, and we want to provide the CNV change for the segment CNV. So let's look at the payload that I have set up here. So this would be seg payload. And similar to before, chromosome is 2, length 100,000, but here we have greater than or equal to. So instead of equal for the operator, which we used before, here we're using greater than or equal to. And then format is TSV, and size is going to be 1,000. There are going to be more, but I limited us to 1,000 just so that we could look at the first 1,000, and we don't, it doesn't take too long. So let's go back to our query here, and first we're going to add our payload to this query. We're going to use the segment CNV's endpoint. We're going to go ahead and run that. And here we have, oh, one more thing. If you notice, I didn't actually add any fields. I'm just using the default because that will give me all of the segments up to 1,000 because that's all I asked for. And it will give the CNV change. So if you're interested in just the CNV change, you have loss, gain, you're only going to see loss and gain. But if you are interested in the CNV change 5 category, you will see loss, gain in addition to amplification and homozygous deletion. So you'll see those in your output. So this doesn't give all of them. It'll just give up to 1,000 because that's all I asked for. But yeah, if you make the number much higher, you can see all of them. And then for the last endpoint, the segment CNV occurrences endpoint, we want to know which cases have segments in chromosome 2 that are at least 100,000 base pairs in length and underwent an amplification. So the same query as before, but in relation to cases, which patients have this. So one last time, we're going to look at the segment occurrences payload. And Same as before, you'll see that the fields are just a little different because we're looking at the occurrences endpoint rather than the segment endpoint. And yeah, value equals 2, greater than or equal to 100,000, and value equals amplification. And now for the fields, I want the same things as before, but I'm listing out the ones that are totally necessary. So um, we have the case, the primary site, the disease type, the CNV length. We just maybe want to validate that our filter worked, and then the CNV change 5 category. It's going to be a TSV, and we're only going to look at 1,000 for now. So segment occurrence payload, and then change the endpoint name. So I'm going to go ahead and run that. And as you can see, similar output to last time, except each of these is now related to a case. So that's the end of my demo, but there's a few things I do want to bring to your attention before we get into questions. One is that if we go into the documentation site, you can get there at docs.gdc.cancer.gov. And if you're interested in the tool we just showed you, if you go to Data Portal and you scroll down to copy number segment, you can see documentation for the copy number segment that Shin demoed. If you're interested in the pipelines, if you click on data here, and if you were to scroll down to the copy number variation analysis under bioinformatics pipeline, you can see information about the workflow type that Zenu presented. And if you're interested in more information about the API, you could click on API at the top here, scroll down, and we have information under data analysis, and then copy number variation and copy number segments. So here they are on the right in the contents here. And lastly, if you have any questions outside of this webinar or later, feel free to email my team, the support team, at support@nci-gdc.datacommons.io. And that's the end of the presentation, so we do have a little time for questions. So let's take a look at those. Okay, so I saw that there were a few questions asked during Shin's demo. So Shin, I want to bring some of this to your attention. So for the first question, what is the data/tracks being presented in the PAX5 illustration? So where does that data come from?
Yeah, let me see the question again. Which question is it?
What is the data/tracks being presented in the PAX5 illustration? So when you did a demo with the PAX5 gene.
Yeah, there are 2 tracks. The first one is the quote unquote copy number tool, right? So it shows all the copy number segments from GDC over the current region from the current cohort. The second one is the gene model, the gene code gene model.
All right, thanks, Jen. The second question, and this is in relation to your demo because that's the time stamp that I see. What disease are these seen in? So,
And you can see that from the sample summary by clicking on the sample text label on the left. You'll be able to see that.
all right, thanks, Jen. The next question, instead of selecting by gene, can the visualization tool focus on specific disease samples?
I also show that you can in the sample in the disease breakdown panel, you can select a particular disease, particular site, organ site, particular race group, etc., to show the subset. The other method is to in the GDC cohort builder, you build up the cohort that you want to look at, and then the CNV tool is cohort centric, and it will only show the data from that cohort.
All right, thanks, Shin. And now getting to uh the API part. Um, yes, I think I totally forgot to say underscore mapping when I was typing that out, but you seem to have figured it out, so thank you. Um, let's see. What if I want to get the CNVs over a particular non-coding region slash locus? Can I specify coordinates in the payload? Um, so this is all gene level, so this is so I don't think you will find the non-coding region in there. Um, in terms of actually finding those in the bio file, in the actual files themselves, because I don't believe they'll appear in the API. But Zenu, do you have any information on whether or not those will be found in the actual um in the files that are downloaded from the repository?
Um, yeah, all the segmenting across the whole genome are available in the downloaded files, including um, different categories of neutral, loss, and gain. Um, does that answer the question? Or
Yeah. So what I will say is that you will need to look at the segments rather than the gene level copy number because the gene level is based on the gene model. Is that correct, Anya?
yeah, so um, the segments integer value segments is the actually was the input of the gene level. So we simply just do a overlapping of the gene range to the segment range, and the genes will inherit the copy number from the segment. If you are looking for a location that does not belong to a range of gene defined by the gene model, then the segment file is what you should look for.
Thanks, Anya. So let's see here. I got another question. It's a bit odd to look for amplifications in TP53 since it's a tumor suppressor, so deletions would be more relevant events. And I totally agree with that. I just kind of randomly picked some genes and put them in there, but correct, you wouldn't want to. It would be weird to look amongst cancer patients to search for extra tumor suppression. I do agree with that. Um, so the next question, does GDC offer any analysis or visualization of CNV in spatial omics data? Um, I will say that we do not have that data at this time. I can't say anything about the future, um, but yeah, we do not have any spatial data at this time. Um, let's see. Does GDC offer any analysis or visualization tools for CN? Oh, that was the same one. I'm sorry. Um, Here we go. So let's see. Yes. So just to reiterate what we were talking about before, Shin did mention about CNVs in an intergenic region. So the segments that he was showing do overlap intergenic regions, but the gene level copy numbers will always be related to specific genes in a specific gene model. Um, and then oh, can you point me to where I can find the segment data to download? That's a good question. So, yeah, I did want to go over this if we did have some time, but since somebody asked, we can take a look. So, if you go to repository, and I would recommend building a cohort before, just so that you would have the information for your specific um, uh, cases of interest. But I'm not going to do that here. So, if you're interested in the segments, you just click on repository. Go down to the left here and you'll see data type is copy number segment. And if you click on that and scroll a little bit more down, you'll see workflow type. And the copy number segment is offered from DNACopy and for GATK4 CNV, just kind of depending on which project you're looking for. I'll say DNACopy that has all already been run now, so you won't. This number will never get bigger. We won't be getting any more DNACopy data, to my knowledge. But GTK4 CNV, this is an ongoing process. So as new projects are have whole genome sequencing data generated, um, you'll see this increase as we do more data releases. So yeah, these are where the segments can be found. And one more thing I did forget to mention before, so I'm going to unclick copy number segment and just click on copy number variation in general under data category. This is a more general field. Um, as you can see, we have many data types, including like the raw intensities, which are like the cell files, masked and copy number segment, the intermediate analysis files. Everything here is open access except, let's click in controlled here, except for certain, um, except for the cell files and the intermediate analysis that result from GTK4 CNV. So that is that you need dbGaP access to get that. And if you scroll down to access, you'll see open and controlled. So if you'd like to filter for that, you can go ahead. Next question, can the visualization tool be used on our own data? The GDC does not offer that yet, but I guess Shin, I'll open that up to you. Is that something that's through, through St. Jude or something someone could do?
Yeah, that's a good point. It depends. I think it's definitely useful in some cases. User want to bring in their own data to have it compared against the GDC data to have them show side by side. That's definitely a good point. We may be have some convenient features so that such as we show simple UIs that people copy paste their copy number data and in the current locus and show that have it compare against the GDC data. That's possible. Yeah.
All right, thanks, Shin. Let me go look through the questions real quick just to make sure I didn't forget any.
I think there is a question asking about which one is better, segment mean or absolute segment value. Um, um, I think segment mean is definitely an like C value. I personally will prefer using the absolute integer values. However, there are two caveats. First is like if you're using some old tools, for example, if you are using GSTIC to derive like a frequent copy number change area in a cohort, it's only compatible with the segment means. And second is, as mentioned, like absolute segment values consider purity and ploidy. There are a lot of modeling, and normally the same data will result in multiple potential models, and one of those will be used to derive the segment you get, but it has possibility that the model being picked is not the right model. That's why copy number inference is very difficult, and a lot of people, the expert really want to do like manual review of some of the samples with uncertainty models. That's what they did. But in general, the reason I like absolute values because, as just mentioned at the beginning, like it's really difficult when you use segment mean. You cannot easily distinguish a low purity sample, but with a drastic copy number changes in the tumor, with a high purity sample with a lot of changes, with less changes. It's very difficult. And also segment mean is kind of noisy. That's why I personally like to use absolute copy number values if it's available.
Hey, thanks, Zenyu. And I totally missed this question. Apologies for that. I mean, I think Zenyu, you actually touched on this a little bit, but the question is, I've heard with ASCAT that it has to be run several times for the same input data to determine the right estimates for purity and ploidy. It is likely the first run result is unreliable. How does the GDC ASCAT workflow handle this issue?
Yeah, that's also related to the multiple model purity issues. And some people using another tool to derive the purity and let it feed into the input of ASCAT. Um, that's part of the reason when we initially adopted ASCAT 2, later we also roll out the ASCAT 3 and absolute data, because ASCAT 3 has been more carefully curated by the author of ASCAT, and the absolute leftover data that's for the SNP6 is definitely a lot better, because I think there is a community of CNV expert. They carefully curated the possible model selection. Yeah, so you're definitely right. Those copy number inference, especially modeling inference, model selection, could sometimes end up to the wrong model, and that will impact data quality. From the GDC perspective, we run a lot of data. We don't really do any manual curation. Everything is done in an automatic fashion. So that's why we rely on community expert to provide the best curated data set.
All right, thanks, Daniel. It looks like we're a little bit over time, but does anyone have any last minute questions? All right, well, if not, I'd like to thank everyone so much for joining us for this webinar, and hope you all have a good Monday. See you later.